It is my great pleasure today to introduce our speaker, Professor Smita Krishnaswamy. Smita Krishnaswamy is an associate professor of computer science and genetics at Yale University. Smita's lab works on fundamental deep learning and mathematical machine learning methods for accelerating discovery from biomedical and neuroscientific data. Her work features breakthrough methods for data denoising, MAGIC, visualization, FATE, dynamics modeling with trajectory net and MyoFlow, comparative analysis with MELD, optimal transport with diffusion EMD, and multimodal integration with MAGANs and SCN GANs. From datasets in neuroscience, psychology, stem cell biology, cancer, immunology, and imaging. Smita obtained her PhD from the University of Michigan in computer science, and Smita's work has won several awards, including the NSF Career Award, Sloan Faculty Fellowship, and Blavatnik Fund for Innovation. So it is my great pleasure to hand it over to Smita. And before I do that, I again want to welcome everyone and remind that this seminar is co-sponsored by the P41 Funded Center for Mesoscale Mapping, housed in the Martinos Center. And without any further ado, I hand it over to you, Smita, and thank you so much for being with us here virtually today.
Thank you, and thank you for the invitation. Today, I'm going to talk about some work from my lab that is more recent. In the past 2-3 years, we've been focusing a lot on both inferring dynamics of cellular and behavioral systems from the respective data sets, as well as characterizing these. Characterize noisy dynamics. It's very important to keep track of the actual signal and the sort of shape of the signal and how it evolves. And that's where we bring in data geometry and topology. So I'll show you how we develop these methods by combining data geometry topology together with deep learning. I hope, oh, what happened? Okay, sorry. So the fundamental kind of question that'll be the theme of the talk is, so we have all this high dimensional, high throughput data, whether it be cellular level data in the form of single cell RNA sequencing, or another data modality that I work with quite a bit, fMRI data, which shows brain activation, signals that are also time varying, and how can these data sets help us discover insights into biology. And what all these kinds of data sets have in common is that they are very high dimensional. Also, they're very high throughput, and they could have complex dynamics that are hard to describe canonically. And so there's several challenges that are related to actually fundamentally representing these data sets so that we can get insights. And this is really what deep neural networks are very good at. They're very good at learning multiple types of representations. And we will show why we use representations that are specifically informed by geometry and topology. So some fundamental challenges that you might want to overcome with this data is, as I said, there might be a lot of noise in the data. Biomedical and specifically neuroscientific data has very high degrees of noise, but there's some kind of structures in the data. I'd like to say temporal and geometric structures that help us recover from the noise. I also mentioned that these data sets are very, very high dimensional. So you're measuring every voxel in the brain or every transcript in the cell. So there's a need to put this into lower dimensions, so that we can interpret what's going on. And we have methods that preserve data geometry that help us do this. And today I'm talking also about how even if you don't have dynamics, the underlying entities in biology are always dynamic systems, right? So we're always evolving, changing, growing. And so how do we put the dynamics in? And that often helps us with gaining more insights into a system. So one of the things I'm going to talk about is at the cellular level, how do we reinfer cellular dynamics from static snapshot data, whether it's single cell or developmental data that could have single cell as well as ATAC-seq. And the idea is we want to understand what would have happened to a cell if we had been able to watch it the same way we watch regions in the brain. And so what we want to get is continuous dynamics of it. And then once we get the continuous dynamics, what we want to do is we want to characterize the dynamics, because often it's these dynamic changes in cellular state or signaling states that confer signatures of disease, and that can actually be quantified. So here is a T fate or temporal fate, which is a method we developed adapting fate to the temporal dynamic setting that shows you different kinds of dynamics. So dynamics where there's a lot of jitter followed by a loop, or continuous looping dynamics. And there's ways we can quantify and classify these dynamics and even recover parameters of the underlying systems. So that's a little bit what we're going to be talking about. But what is the main idea behind trying to recover signals from these high-dimensional, high-throughput data sets? The idea is, even though you're measuring maybe what seems like 20,000 dimensions and you feel like you have a network that's very, very high dimensional, the number of processes going on in cells is not that many. It can be a dozen or 2 dozen. So this brings the intrinsic dimensionality of the data much lower. And if you were to focus on, for example, learning these latent variables instead of the 20,000 genes, you might get a better idea of what's going on. So there's an underlying latent space that's much lower dimensional. It's the same theme in the neural activity data. Neural activity data, we're at some level of granularity measuring how neurons communicate with one another. But because they're all communicating with each other, because they're all talking with one another, there is something of the same message that's going through. So neurons have a lot of mutual information between them, which lowers the dimensionality of the activation trajectories. So this kind of thing that I'm saying is well captured in a mathematical assumption called the manifold hypothesis. So the manifold hypothesis, and it has become a pretty famous hypothesis in machine learning because it allows us to tackle all kinds of problems, particularly from biomedical data, is the idea that there is some structure in the data due to the redundancy or the relationship between features, or the mutual information between features that restricts the state space. And really, the observations you're seeing are samples from this manifold that have some noise added. So if you denoise and try to learn the shape of this data, you'll get quite a lot of information. To give you another illustration of this, if you just, let's say you just had 3 features. If all the features were uncorrelated, there's no shape here, right? But the minute they're correlated, you get shape. And by learning the shape of this data, you also learn the correlation structure. And that is kind of the idea of the manifold hypothesis, that the data lies in a lower dimensional shape, and that if you have enough samples of this shape, you can actually learn it. And once you learn this shape, you can restore noise, you can figure out new intrinsic latent dimensions, such as this dimensions in which to visualize or represent the data, and a lot of the problems kind of become automatically solved. And this is kind of a theme that our lab has been, developing and pioneering since its inception in 2015. And so how do we learn the shape of any kind of data? Via the points that you observe. So let's say each of these points is a point in just 2D space. One of the ways of learning this kind of shape is to turn this into a graph. So how do you turn this kind of point cloud into a graph? Here's one commonly used method. So if you go with the example of cell by gene data, these are observations and these are features. You would simplify that into a distance matrix where you're just taking distances between these rows, row I and row J. And then these distances are not necessarily reliable because they don't lie in this intrinsic shape. And so what you do is you localize the distances into an affinity by using a kernel like this. This is called an exponential kernel, and we just put this distance into the kernel and it inverts it so that small distances have high affinity and large distances have low affinity. And this sharp drop off is important because it'll give you something that more or less preserves local relationship. And now you can see this graph is sort of like a nearest neighbors graph. And this graph allows you to basically walk through the data. And walking through the data to learn its shape turns out to be a very important and deep concept in manifold learning. That is just coming together right now when people are finding its need in machine learning. So the way you actually walk through data is by creating what's called a data diffusion operator. So you take this affinity matrix you created like this, which I'm calling A here, and you row normalize it. And this gives you a transition operator that shows you how to walk on this graph. And this Markov matrix, what you'll notice is the affinity matrix could even still have some connections that are cutting across the manifold, but the Markov matrix can be powered, and the powering makes it strongly go through, it makes it go through T-step walks. And these T-step walks have a very high probability of remaining on the manifold. And that's called a diffusion operator. So of course, you can also define a diffusion operator for data that's already given as a graph. So if you have a given graph, or if you have like an anatomically determined graph where you're just connecting adjacent components in anatomical fashion, you can still just take the graph and normalize it, and you'll get this kind of transition operator that you can then use to denoise and learn about the manifold. The interesting thing about this data transition operator or data diffusion operator is it's the discretization of heat flow on a graph. Basically, if you have a unit of heat and you let it spread on the manifold, and you have an associated operator called the Laplace-Beltrami operator, then many people have shown that this kind of data diffusion operator converges to the heat operator. And there's very fundamental connections between flowing heat on a manifold and finding manifold intrinsic distances, which turn out to be the key to representing this data. So with that kind of introduction to our kind of graph-based methods for manifold learning and the idea that data has shape, I'm just going to say that in general, we combine the geometric notions of distance on a manifold, which we find using diffusion on a manifold, and topology, which characterizes sort of these shapes of these kinds of graphs, together with deep learning. And this has been kind of a formula that has worked for us to produce denoising, visualization, and other sorts of methods. But today I'm going to start talking about dynamics. So the first idea is, how do we learn cellular dynamics? And we have some projects in neuroscience where we're trying to learn developmental dynamics, ordinary developmental dynamics, as well as perturbed developmental dynamics. And this is actually when you don't even have dynamics, and we feel like it's important to imbue the data with the dynamics anyways. And so here we have a neural network that I'm going to present, that we presented at NeurIPS like a year ago, not the one that just happened, but a year ago. It's called manifold interpolating flows. And the main idea is to actually learn a manifold intrinsic latent space based on the diffusion operator, and then take the data here and flow it using an neural ordinary differential equation. This is a neural network that gives the derivative, and you send it through an ODE solver, and it can give you dynamics. So I'll talk a little bit about how to do this. So here's some developmental data. So these are human embryonic stem cells, and they're developing into many lineages, with some of the most prominent ones being neural progenitor and neural crest right there. But we have static snapshot time points of this kind of data, so 5 time points. And we want to learn how cells develop along that. So there has been a lot of effort at trying to understand cellular dynamics from static data. So there's some methods like RNA velocity that try to give the arrow of where the cell will move next. Some try to match between time points, like, oh, this cell could have come from this cell, but you're not really sure what happened in between. So what we're actually trying to do is recover the entire trajectory. And we want something like this, like a smooth dynamic trajectory. And we started doing this back in 2020, where we first published a method called TrajectoryNet that was older than this method. And so in order to do that, we first understand that if a cell is moving, it has to be moving along the cellular manifold. Otherwise, you'd have samples outside the manifold, or the outside would be part of it. So we try to put it strongly into manifold intrinsic coordinates. And the way we do that is we penalize multi-scale diffusion distances based on the rows of the diffusion operator I showed you between these cellular measurements. So why do we do it multi-scale? The reason we do multi-scale is because multi-scale diffusion distances at a population level are equivalent to an optimal transport distance. So this gives us a very cool connection. Also, we're not quite sure what is the right number of steps to diffuse to get the correct manifold distances, and then this avoids having to select that. It gives you a dyadic scale. And so in an earlier paper that we had, what we were able to prove is that if you have multi-scale diffusion distances between signals on your graph, which could represent cells from time point 1, cells from time point 2, and cells from time point 3, this kind of multi-scale diffusion distance is equivalent to an Earth mover's distance. So the Earth mover's distance is a distance between populations when you're flowing one population to another. What is the amount of work you have to do? If you think of it as mounds of dirt, how hard is it to change one mound to another? But this turns out to be a very interesting kind of Earth mover's distance where you're not moving on the ground, but you're moving on the manifold. So that's an Earth mover's distance with respect to the diffusion ground distance. So what we do is we then train a neural ODE in this space. A neural ODE network is a network that produces a derivative, and that derivative is then integrated via an ODE solver to the next time point. So what this allows us to do is take time point 1 and come up with a flow for time point 1 such that, or rather 0, such that at time point 1 at a population level, it matches this other population. And then we penalize these two populations with optimal transport. So with the multi-scale ground distance, which is an optimal transport distance. So basically we come up with a flow for each and every single cell. So every single cell gets a continuous dynamic flow that we come up about how it transitions from time point 0 to 1 to 2 to 3 and on and on. And the only penalty is that at the population level, it matches the populations we've actually measured at those time points. And there's one other very important regularization, which is to penalize the magnitude of this derivative that comes out of this network, because we don't want squiggly paths that are implausible. We want the paths to be as smooth as possible because we don't have any information at higher frequencies than this. So when we do penalize the magnitude of the derivative, we actually get, and we penalize at the time points we have with optimal transport, then we get something called dynamic optimal transport, which was a framework that was developed in 2000, hadn't been used much. The idea of dynamic optimal transport is that not only do you move your dirt using an optimal transport plan, but the way you move it is also optimal. And so dynamic optimal transport will take points using shortest paths to the destination, not only with minimally modifying these, but the plan of taking them there. And so you don't have this circuitous movement. So if you're going from a Gaussian to an S, you move them directly to the S instead of sliding down the S like this. There are other things that are happening in cells. What about cells dying or proliferating? So this too can be taken into account with optimal transport if you allow for what's called imbalance. So if it takes too much effort to move a cellular population, we won't do it. We'll just kill those cells. And if you have something like RNA velocity, it's very strong signal in your data. Sometimes it's not a strong signal, sometimes it's very noisy. Then where it's available, you can also penalize to agree with it. So at the end of the day, we can get these kinds of dynamic trajectories and that show how these cells, which were all human embryonic stem cells from day 0, how they had slight differences between them that allowed them to develop into different lineages. And especially these cells from here developed into neural crest and neural progenitors. We've also used this in disease systems. So we've used it on different kinds of cancer where we can tell how cells can go from a benign kind of cancer to a more metastatic transcriptional profile. And one of the interesting things about these is you get a trajectory for every single cell, and I haven't visualized all of them, but it's very clear that there's at least 2 types of trajectories. There's a metastatic-like trajectory and a benign-like trajectory. And then what you can do is, because these trajectories are summarizations of your high-dimensional state space, you can use the decoder of that autoencoder to go back to the gene expression space and see how the dynamics of each gene are very different between tumor-forming and non-tumor-forming lineages. So we could do that in disease. And this gives you kind of a dynamic way of also trying to say which gene might be upregulating the next one, because now suddenly you have timed gene trends. So initially, we used Granger causality along with these time gene trends to come up with networks that were driving these processes forward. But recently, we've actually also switched that to a dynamic framework, and I'll show you that. So a little bit about our ongoing work in neural development. So we decided to go deeper into the neuronal lineages you saw in the same stem cell data. And our stem cell collaborator generated these for us, where we were focusing on the neural lineages. And we saw the precursors of several different types of lineages. And now what these gene dynamics tell us is how you push towards one cell fate versus another. And what's pretty interesting is that it can tell you that some of these dynamics are very distinct in one cell type than another. And you could potentially offer a timed perturbation to change the lineages potentially. And so that's where we're going with that. So as I mentioned, what we were very interested in once we got these timed gene dynamics for each cellular trajectory was to understand the network driving, the underlying gene regulatory network, if you will. And the same thing can be said if you see a dynamic that you didn't have to infer. For example, you had brain activity dynamics from fMRI data or calcium signaling data. We could try to infer the underlying regulatory circuit for them without knowing it in advance. And what we're doing for that is we're trying to learn the connections of the graph itself using graph attention and a graph ODE network called Routini that we recently published in a conference called Learning on Graphs. So here was the idea. We have now the dynamic trajectory traces, whether these trajectories are from the cellular dynamics that we just inferred or brain dynamics, brain activity dynamics that we've already measured. And what we try to do is we try to get this graph neural network to dynamically change its connectivity over time to be able to predict the dynamics in high dimensions. So each vertex here pertains to a gene, for example, using the system that I just talked about in the previous story. And what it's saying is this gene is paying attention to these two other genes in order to change its regulation. And that could mean these two genes are connected to these other two genes in a regulatory fashion. And it doesn't tell you the exact mechanistics, but just that there's a regulatory effect because it requires this attention to predict the dynamics. So in order to do this, we had to design a very specific kind of attention. This was attention over time that allows you to accumulate effects. And so you're paying attention to your neighbors, which could be upstream regulators over a time lag. And there's autocorrelation. There's already some amount of the substrate of the present network built. And it also supports hysteresis. And the way that the hysteresis is supported is that these kinds of attention coefficients themselves are looking at time traces of information. So the attention basically designed in this way gives you a time varying graph or network. It supports looking over intervals of time, like letting one signal accumulate. So it can model, for example, a firing system where you integrate and fire. And then it outputs an instantaneous derivative to predict the dynamic. So we use this attention model pretty strongly to feed into the ODE solver that we have of this graph ODE. And we penalize by how well does it match our signals, as well as how well does it stay close to a prior network if you have a guess of a prior network. So one very interesting thing about this is not only does it learn time varying networks, but it also allows for simulating perturbations. Perturbations can be simulated by altering node features. So you can, perturb the feature of a node, and you can see how that can cascade to the next node, to the next node. And if a time perturbation is applied, then its effects also propagate in time through the network, and you can predict that. But what is a very interesting possibility is as we get more perturbation data sets in these domains, whether it's cellular or brain activity data, you can adjust the training of these networks to better predict time perturbations far out in time. So that's an intriguing possibility for which we don't quite have all the data in all the systems. But here's an example. And so in the neural crest and neural progenitors lineage specification example I gave, the blue is the neural crest and the brownish color is the neural progenitor. And we can have the dynamics of specific genes that are involved in their development. And we can knock out other genes and see what happens when you knock out other genes. So for example, if you knock out TFAP2 in silico again, and you do it at this particular time, then what happens? So it seems like the SOX9 goes down immediately in neural crest, even though it had started to become higher. So that could prevent that lineage. And what's interesting is you might be able to do multiple knockouts, which gets combinatorially very expensive to do in real experimental systems. So again, as I mentioned, we tested these both on integrate and fire type of networks using the NEST simulator, as well as gene regulatory networks for specifying cell types and differentiation. And in both cases, we found that our methods are performing well at understanding how the network is changing. And I will say that most of our comparative partners give a static network prediction. So this isn't exactly an apples to apples prediction, whereas these are all giving one static networks. Ours actually gives a dynamic network. And to make the static comparison, we gave we averaged over these. So ongoing work would be to also take into account spatial or anatomical information to make these networks more precise and microenvironment dependent. So now I'm going to move on from the idea that we have to infer networks, infer regulatory, potentially simulation-oriented system, to what do we do now that we have these dynamics? So we have Routini can simulate dynamics. We had Miaflow gave us dynamics, or sometimes we already have dynamics, but how do we gain information about what's going on? And here I'm going to talk about the latest addition to our FATE development. So initially we had a method called FATE that captured manifold geometry. Then we made it multi-scale. So we were able to analyze populations with a hierarchical structure like immune populations. And then very recently we made it time varying. And this time varying visualization is good for data that has some kind of dynamic nature. So the whole idea of FATE is that it preserves the geometry of the data because it preserves manifold distances as derived by the diffusion operator, but the actual distance we compute from the diffusion operator is a little different in FATE. The idea is if you had data that was like this, you sampled, this is ground truth, you sample along this, add noise, rotate it into very high dimensions, and you tell dimensionality reduction algorithms to capture it, they will capture different aspects of the data. This is kind of like the blind people looking at the elephant. Some people capture the tail, some people capture the trunk, whatever it is, because we're trying to take a very high dimensional system and capture it in 2 dimensions. But we see that some of the things that have been captured before are things like maybe cluster structure, but this is shattering the overall geometry. Or it could capture some of the trajectories, but not all in 2 dimensions. It could make a linear projection, which actually is the opposite. It kind of gives you a global picture, but doesn't give you the local branching structure. So what we wanted to preserve was a different aspect, which is we wanted to preserve the manifold geometry, as in the manifold distance going from one data point to another via the data. And so in order to do that, again, we go from data to distances to affinities to these diffusion probabilities. But here, each row of this matrix contains a probability distribution we realized. And most of the other manifold distances, like the ones I showed you before, are just taking a Euclidean distance between them. Instead, to capture the most amount of information, but here, we decided to use an information theoretic distance, which uses this log factor before computing the distance, and then embed it with multidimensional scaling. So here, this divergence that you take up weights the effect of far distances. So for example, if you had this data point here, it would not only be contextualized by things close to it, but by things that are farther away from it. And if there's a population way here, it could influence it as well. And that's the idea of if near and far things contextualizing because of the log damping factor. So the overall effect of the steps is that step 0 denoises the data because you get these diffusion probabilities and you can power them, and the powering gives you the clean manifold distances. Then each cell is represented as a t-step diffusion probability to every other cell in the rows of the diffusion operator. And then you compute this divergence between these probability distributions that really says if you started at this point versus this point, what would be the difference in the random walks that arise from there? And then you preserve that information in low dimensions. And I'm going to skip over a little bit of this in the interest of time. One really important thing that we've figured out is that fate has a deep connection with heat diffusion. There's a formula called Varadhan's formula that connects manifold distances to the heat kernel in the limit st goes to 0, and you notice this log factor appearing here again. And recently we wrote a paper at NERPS called a heat geodesic perspective on nonlinear dimensionality reduction that talks about how intrinsically this is what a lot of nonlinear dimensionality reduction methods do. So this is what fate does. Some of you may be familiar with diffusion maps. That's different than fate because diffusion maps eigen decompose the diffusion operator. And if you eigen decompose the diffusion operator, what you get is each eigenvector is roughly one trajectory. And so these trajectories become separated. And so it's better as a method of clustering these trajectories. Whereas if you visualize these diffusion components here, you see they're forming different arms. So they actually work quite well together because fate gathers them all into one plot, and diffusion maps will highlight particular trajectories. So again, I've been kind of flashing to that stem cell developmental system that I'm returning to. And the idea here is that I already oriented you that these were human embryonic stem cells and they're developing into different lineages. But you see, you cannot see that in most of these other visualizations. And that's because they're not keeping the relationships between points, and therefore they're not really keeping the dynamics together. And so PCA, you can see globally does, but it cannot denoise in these nonlinear directions. TSNE or something like that only retain cluster structure will shatter this. And diffusion maps, they, on average, each diffusion component only shows one trajectory. So now the question is, how do we extend fate into data where we have the temporal dynamics that we figured out? For example, in brain activity data or fMRI data. So what we do is we take each time point of measurement that we have and we connect the time points both based on the original fate view of, comparing all the activations at the time point, and a temporal view that gives you an autocorrelation kernel that says similar time points have to look similar. And if you combine these two, this temporal kernel with the fate kernel, you combine them basically by multiplication, you get a new T-fate diffusion operator. And this operator diffuses sort of both in time and in space. And then this operator does a better job of preserving the manifold when you have dynamic traces of your system. And this shows the difference between these, and it is particularly useful in very high noise settings which neuroscientific systems have. So things like PCA don't retain this kind of U-shaped structure. As if you get even more noise than fate can handle, fate is already denoising, then you would need T-fate because it also uses temporal autocorrelations to denoise. And these were used on fMRI data of single subjects watching movies. And you see that if you embed the brain activity and what happens to their brain activity over time, from time 0 all the way up to, this time nearly 2000, then basically using almost any other technique, you get this inchoate blob. Whereas in T-Fate, you see that the brain activity follows a continuous trajectory as the movie evolves and the scenes of the movie change. You can get clear segmentation based on how this trajectory proceeds. For example, here you can tell this movie has like two sort of very different looking halves of it. And what was interesting was we wanted to check if these boundaries corresponded with the boundaries people would draw by watching the movie. And so that's kind of what we did. We looked at other models people had for these kind of boundaries. So this is an HMM event boundary model. And we looked at the correlation between the pattern and the boundary. And we see that T-Fate routinely has the highest correlation between movie segmentation and the segmentation of the visualization. And we don't see that in PCA, UMAP, Fate, or even Fate. So these high bars are showing that the dynamics offer the segmentation or the different regimes of the stimulus. So this was very interesting to us, and we wanted to do more in as far as characterizing these kinds of dynamics and breaking them up into different segments and recovering stimulus features. So recently we went another step further and developed a geometric scattering homology trajectory. And what this is trying to take into account spatiotemporal dynamics. So T-Fate takes into account temporal dynamics and brain activity, but this puts the brain activity onto a spatial substrate and takes into account the spatial as well as temporal localization of these signals. So we've used this most extensively on calcium signaling data. So initially, when we developed this, we actually got some interesting calcium signaling from epithelial cells. So these aren't even neurons, these are epithelial cells, and they're signaling calcium, but people don't know why. Why are these epithelial cells trading calcium signals? And one thing it might look like these signals are somehow random. They look like random flashes. You know, what's going on? Is there any regulation or coordination? So what we did was initially we created a graph out of the epithelial cell tissue. You could also do this with neurons or voxels. And then we created a descriptor for the signal as lying on that space using graph signal processing. And then we took these descriptors and put them into T-Fate to try to understand the dynamics of these signals as they play out on this spatial substrate. And finally, we can just characterize this kind of trajectory with topology, and that gives us the final characterization of this entire dynamic. So the graph creation is if you have this kind of morphological information, it's between adjacent cells. In fMRI data, we often just create the graph between adjacent voxels. And then the signal is considered some kind of feature on the graph, okay? And then the idea is that you can capture features of these signals using graph signal processing. For example, a famous transform, Fourier transform, has been moved into the graph realm, what is called a graph Fourier transform. And that's to load these signals onto the eigenvectors of the Laplacian, or actually the diffusion operator has the same eigenvectors if you normalize it correctly. And you get a frequency domain representation. So that's not quite what we did. But one interesting thing we used was a frequency domain filter. So if you apply the diffusion operator, especially a powered diffusion operator, onto the data, you get a low pass filter. Okay, so this is powered T2, lose some high frequency signals. T5, you lose a lot. T is 100, it's a very high low pass filter. So using these, you can create a cascade of what are called diffusion wavelets. So you can create a wavelet transform. So there's a Fourier transform in graph signal processing, and now there's a wavelet transform in graph signal processing. And the wavelet transform consists of these low pass filters and high pass filters that are created by subtracting the signal from the low pass filter. So what we do is we take various scales of this diffusion by powering to different dyadic powers. We take subtractions between them to create wavelets, These are called diffusion wavelets, and they've been defined before on graphs by actually Koifman, who also invented diffusion maps. So we took these diffusion wavelets, and what these diffusion wavelets do is if you have an overall signal, a signal right here, which is a composite of maybe a slow moving signal and an oscillating signal, the wavelets can isolate different frequencies of the signal, whereas just the diffusion just sort of smooths it. So what we do is we take the signal and we alternate wavelets with this kind of modulus nonlinearity, and it could be a ReLU. We've recently used ReLU here. And this creates what's called a scattering path. And this kind of scattering path can capture a lot of different information about the signals. So you can place signals on each vertex or whatever signal you have in your calcium signaling network. And you can take these signals and use their scattering coefficients, and you can get a scattering coefficient for the whole graph by aggregating all the node vertex signals like this. So it can be invariant to the size of the graph too. So we've created a trainable version of geometric scattering where we create these different scales of diffusion. We learn the scales that we create the wavelets, and then we create the aggregation module, and it gives us a set of coefficients for the entire graph. So these are called scattering transforms, and the interesting thing is they can be made deep. You can take these scattering coefficients and then use them as signals in the second frame, and you can create a deep network that's much like a neural network. And that's why we were interested in integrating into a neural network. And it turns out to be much more powerful for capturing what an entire signaling dynamic is spatially. So next, we just take these scattering coefficients that we got at each point in time, capturing the entire anatomical signaling pattern, and we just put it through, initially we put it through FATE, now we're putting it through T-FATE, but in this picture, I think we actually put it through FATE. And we see what these calcium signaling dynamics in the epithelial cells were like. They were actually very smooth and kind of cyclical. They were not random at all, right? And how do we capture that quickly with a signature? We perform topological data analysis. So this is just a Vietoris-Rips persistence diagram. So for those of you who aren't familiar, Vietoris-Rips persistence diagrams compute topological features like number of connected components, holes or cycles in a shape, and they do it on a graph. So for example, you grow this epsilon ball and you see how many connected components and when they appear and when they disappear. And you can keep doing this, and then you could get a cycle, and you can count cycles with a different color. And that's what that persistence diagram I showed is. So for example, now we see in the basal epidermal layer, we had a very smooth dynamic of signaling, whereas in neurons or neural visual cortex, we have a very jumpy dynamic. And that signature is available in the persistence diagram. So now we could just feed this into a machine learning algorithm and classify, so for example, the source of the dynamic. And that's really what we've been doing. So we've collected very interesting experiments. So for example, in this paper, we perturbed the dynamic by stalling G1, enriching with drug, and we see every time we perturb the system, somehow the dynamics get perturbed too. And so we can classify these kinds of dynamic changes. I'll show you in a second. And the way we can classify them is by first coming up with these persistence diagram topological descriptors. I'm going to skip this slide. One thing that we see is which parts of the regulatory network are necessary for this dynamic. Because if we remove, for example, a potential molecule that's necessary for long-range calcium signaling, you see some disruption of the dynamic. So then we've also seen calcium signaling in a mouse epidermis, and we see the same effects. When we apply different drugs, we get different effects, and those can be classified. And the way we classify them is we just take the output of GSTH. This is our model, and these are different ablations of our model using PCA, TSNE, using an RNN instead of scattering or the homology, and we get good results, consistently good results with the GST, which is the T-FATE that's applied on top of the geometric scattering. and also the homology at certain points in time. So here's an example of how we would use this in the future. So for example, let's say we were teaching mice to recognize these sinusoidal grading patterns. So then we look at the brain activity and we get these kind of dynamics. Okay, and then what we do is we take these dynamics and you could either take the T-fate or you could take the homology and you can just put it through a classifier. So now we're seeing that these classifiers are performing better than other things like chymograph, Crockers, and zigzag persistence. And in the epidermal layers, we're classifying between drug perturbation and lack of drug perturbation. In the ERK signaling, we're looking at different treatments. And here we're looking at the orientation, spatial frequency, temporal frequency of these grading patterns, as well as orientation, and we classify those the best. So that means that we can actually come up with classifications or even learn model parameters that generate these as regressions using this. So, and that's where I'm going to complete my talk today. So I think hopefully I give you a whole idea on why dynamics are important, why we should try to infer dynamics when we even don't have them. And once we have them, how do we visualize them and eventually use them to gain quantitative insight into the stimulus of origin, what's generating them, generative models, underlying graphs, to really understand a lot of insights about the systems that you're looking at. If you're interested in anything I talked about, most of it's on GitHub here. And then I just really want to thank all my collaborators and people in the different labs in which I belong, calcium signaling data from Valentina Greco's lab, cancer data from Christine Schaefer, Erica Bush, my collaborator from the Nick Turk Brown lab. And if you have any questions, please let me know.